Hi, everybody, and welcome to this week's lecture. And what we're going to be covering for this week is going to be specifically what are nucleic acids, what is DNA, and exactly what is RNA going to be. So, our learning objectives are going to be able to compare and contrast the structures of the nucleic acids, DNA, and RNA. We're also going to try to sketch the DNA double helix. So, we're going to look at the specific structure and exactly why and how that's going to be comprised. We're also going to be highlighting specific parts of the DNA, specifically the backbone. We're going to understand the rule of complementary base pairs, and we're going to see exactly why we have something known as an anti-parallel strand arrangement. So let's go ahead and get started. One of the very first things we need to identify is exactly what a nucleic acid is. So all a nucleic acid is something that's going to constitute the genetic material of living organisms. So this is going to make up basically the instructions on how your body is going to be comprised and exactly what is going to be produced where and how it's going to kind of work and when we need to produce them. So there are going to be two different types of nucleic acids. We are going to have DNA and we're going to have RNA as well. So the reason why it's known as DNA is because it's known as deoxyribonucleic acid. You can see we have our deoxy right here, D, nucleic, N, and then acid over here. Versus our RNA, it's just going to be ribonucleic acid. So I'm sure you're wondering, well, what's the difference between the two? It is going to be a specific sugar that is going to separate them both, and their functions are going to be very different as well. So deoxyribo is going to be the sugar versus ribo is going to be the sugar for RNA. So another thing we need to identify is going to be the location of DNA within cells. So remember, we have two different types. We have our prokaryotic as well as our eukaryotic cells. And remember, there are going to be some major differences between the two, and those differences are going to dictate exactly where that DNA is going to be found. So if you can remember, in eukaryotic cells, remember EU equals true, meaning it has a true nucleus. And just like that, the amount it kind of reminds you that eukaryotic cells are going to have a nucleus, meaning DNA is going to be found within that nucleus. Now remember, we also have the mitochondria, and that is going to contain DNA as well. So in eukaryotic cells, it's going to be the nucleus as well as the mitochondria. Unlike prokaryotic cells, remember, pro equals no, meaning that they do not have a true nucleus. So in this case, that DNA is going to be identified or located in the cytoplasm. So that kind of brings up the question, why is prokaryotic DNA not located in the nucleus? Remember, the nucleus is only going to be found in our eukaryotic cells and not in our prokaryotic cells. So let's go ahead and see specifically the location and the appearance of DNA within eukaryotic cells, because we know they're going to be found within the nucleus, but how are they going to be comprised? What do they look like? And exactly what's going on with them? So as you know, DNA is going to be the instructions for the genome of the cell, which is going to be the entire genomic content. So basically what that means is DNA is going to have everything we need to code for our body. Again, it's going to be the blueprints for everything and how to construct everything. So chromosomes are going to be thread-like DNA structures that are going to be tightly wound. So you can see over here, this kind of X shape is going to be our chromosome, and it is going to be comprised of our DNA. What's going to happen is you can see all these kind of squiggly lines in between. That's going to be all of our DNA here, very, very tightly wound together in order to give our X shape. So chromosomes are going to contain thousands of genes. And genes contain, again, the instructions for producing proteins and various forms of RNA. Again, as we've discussed in previous lectures, proteins are incredibly important, and we have many, many different forms of these proteins, which allow for many different functions within the body. DNA also is going to control all cellular activity by turning genes on and off. Okay, so again, they're going to be incredibly powerful, and that's why the nucleus is typically known as our control center, because again, it contains all of our DNA or our genetic information, which again produces those proteins and allows for the turning on and off of genes. So let's go ahead and dive a little bit deeper into what chromosomes are, exactly what's going on with them, what do they look like, and why do we need them? So in humans, we have about 23 pairs of chromosomes right here, and that's going to give us a total of 46. So the cool thing is you're going to inherit one of these sets of chromosomes from your mother and the other set from your father. So again, you can see that we have these numbered over here, 1 all the way through 22. These are all going to be known as our autosomal cells. I'm sorry, not cells, autosomal chromosomes. So these are all going to code for the body. And then over here, these are going to be known as our sex chromosomes. And again, this is going to dictate the sex. Now, again, you can see each one we have two. We have in position 1, 2, position 2, 2, position 3, 2, so on and so forth. And again, the reason why is because you can get about half from your mom and half from your dad. And then those are going to come together because, again, your mom is going to contribute that through her egg cells and then dad through the sperm cells. And then when they come together, again, 23 plus 23 is going to give us a total of 46, or again, 23 pairs. So again, the non-sex chromosomes, 1 through 22, are going to be known as our autosomal, which are going to code for the body. And again, you'll notice starting at number 1, it's going to be the largest, and then it's going to go all the way down to our 22nd here. And you can see they are going to be decreasing in size. Now, again, the X and Y chromosomes are going to be the sex ones, and these are going to dictate whether it's going to be male or female. Again, females are going to have 2 X chromosomes, XX, and males are going to have XY chromosomes. All right, so now that we've discussed a little bit of those chromosomes, specifically identifying our autosomal, which is our body, and then our sex, which is, again, going to relate to sex, now we're going to see exactly the monomers of our nucleic acids. So DNA and RNA are going to consist of monomers. Remember, mono meaning small, known as our nucleotides. Now, nucleotides join to make our DNA or our RNA strands. And in our nucleotide, over here, you can see we're going to be consisting of 1, 2, and 3 pieces. So in this example, for a DNA nucleotide, you can see it makes or consists of a phosphate, a deoxyribose sugar, and then our base, which we'll go ahead and talk about more in just a second. Now, one thing that's important that's going to be very, very different between DNA and RNA is in DNA, remember, our sugar is our deoxyribose. When it comes to our RNA, this is going to change, and that is going to be just ribose sugar. One other thing I need to mention is this right here, when together, is going to create our sugar phosphate backbone. And these are going to bind with other nucleotides in order to create our iconic ladder shape, which is going to then give us our double helix shape. This is going to be our base, and we are going to have very specific base pairing rules that are going to allow these to line up with other complementary pieces. So let's go ahead and check out our DNA and RNA nucleotides. Over here, you can see we have RNA, which again, we have a different sugar. And then over here, again, ribose, and then over here we have our DNA, which is going to be our deoxyribose sugar. But you can still see that we still have our phosphate group right here and right here. And you can see that we still have our nitrogen bases. Now, when it comes to our nitrogen bases, one thing that's going to be very important is there's going to be a very specific way that these bases are going to line up with each other. In this case, you can see our nitrogen bases here are A, C, G, and T. Now, the way that these are going to come together, and this is going to be specific to DNA, is A is going to bind with T. Now, one way to kind of remember this is if you think about A as in apples, where can apples be found? They're going to be found in trees, T. So A, adenine, is going to bind with T, thymine. Then we can see that C and G are going to go together. So again, C and G are going to bind together. A way you can remember this is cars are typically found in garages, or you can think about it as cars are going to need gas. So again, this is going to be our base pairing rule. Anytime we have our nucleotide and one of the bases is an A, it is always going to bind with T. It will never bind with C or G. It's always going to bind with T. Same thing, T is always going to bind with A. And then the same exact rule is going to apply when it comes down here. C is always going to bind with G, and G is always going to go with C. So again, these are complementary pieces, and they're always going to try to find each other. Now, when it comes to RNA, things are going to be slightly different. You still have our A, C, G, but instead of T, we're going to have uracil here. So again, this is replacing our T. So A is going to go with U, and then C, cars are still going to be found in garages, G. So these are going to be the base pairing rules when it comes to RNA. A goes with U, and then C goes with G. So again, you can see the types of bases here. We do have polymerases, which are going to be cytosine and thymine, and then uracils, and then purines are going to be adenine and guanine. So the major difference between purines and polymerase are going to be these shapes. Polymerase, you can see it's just going to be one right here, versus purines are going to be 1, 2, and 1 and 2. Okay, so that kind of summarizes our DNA and RNA nucleotides. Again, one of the most important things to remember are going to be these here, our base pairing rules. DNA, A goes with T, apples are found in trees, and then C are going to go with G's, cars found in garages, or cars need gas. And then A goes with U, so remember RNA, the major difference is going to be that U, and C is still going to go with G. Okay, so now what we need to do is we need to identify what kind of structure DNA is going to have. Now, DNA has a very iconic double helix structure, and you can see this iconic structure right over here. You can see that it's going to have that twisting or spinning kind of shape to it. Now, this is what it's going to look like when it's all laid out. You can see, again, A goes with T, because remember, we're dealing with D and A, so A goes with T. And then our C goes with G. Remember, apples are found in trees, and then cars are found in garages. You can see A binds with T, C, G, T goes with A, C goes with G, T, A, T, I'm sorry, A, T, T, A, G, C, and then A and T. Now, again, when all these bind up together, remember, these are going to be our bases, and then these squares are representing our sugar phosphate backbone. Again, when we look at figure A here, this ribbon portion right over here is going to be our sugar phosphate. And then the interior portion is going to be our bases. So one really important thing when it comes to this double helix structure is whenever we lay it out, you can see that our purines and polymerase are going to come together. Remember, we have 1, 2, and 1, and they're going to be binding together, and they're going to be utilizing something known as a hydrogen bond in order to bind together. Now, hydrogen bonds are going to be very special because these are going to be just strong enough to keep these together, but just weak enough to break apart when we get into DNA replication. Now, one really important thing is this is going to have a very specific structure known as anti-parallel. The reason why is because you'll notice that in this way, everything is going to be going up or pointing up, and in this way, everything is going to be pointing down. So again, one side is going to be going up, and one side is going to be going down. So again, when A binds with T, C goes with G, you have kind of like this reverse shape going on. Again, anti-parallel, meaning that they're still in parallel, but they're anti going in opposite directions. Okay, so again, some major takeaways from this is that the sugar phosphate is going to be on the outside of the helix. So remember all of these right here, all of these right here, or the ribbon portions right in between here are going to be our sugar phosphate. Again, they're lying on the outside of the helix. And what they're going to do is they're going to provide support for our quote unquote ladder, which is going to be our nitrogen bases, which are going to be our steps. Again, all of the pieces that are binding together in the middle. And again, remember, in DNA right here, A is going to go with T and C is going to go with G. Apples and trees, cars and garages. All right, so complementary base pairing in DNA double helixes. I want to make sure I'm stressing this a lot because this is going to be a very important rule. So again, remember, base pairing is crucial for copying DNA. Again, DNA has a very, very specific code, and only certain things are going to come together. So again, base pairing is critical for copying DNA, specific DNA replication before cell division, and copying the information in genes to make proteins transcription. So you can see over here, we have our double helix. Again, T goes with A, which is going to be a complementary, and then G goes with C. And remember, they're being held together by our hydrogen bonds. Remember, hydrogen bonds are very special because they're just strong enough to keep everything together, but they're just weak enough to be broken apart during DNA replication. And again, remember DNA, the two strands are going to run anti-parallel. So that means we have one way going up and then one way going down. You can see this a little bit more clear in this page that we have these pointing upwards and then these pointing downwards. Again, anti-parallel, parallel, they're going to be both, again, next to each other, anti-opposite directions. Okay, and that is going to conclude the lecture on DNA. Really quickly, just to kind of summarize and reiterate some things, we know that we have our DNA, and we know we have our RNA. We know that in DNA, our bases are going to be A and T, C and G. Remember, A and T are going to go together. C and G are going to go together. In RNA, remember, we're replacing that T with the U. So A and U are going to go together, and then C and G. Remember, DNA and RNA are going to be made out of nucleotides, and we have three major components: 1, phosphate, 2, sugar, and 3, our base. When they all come together, it produces a very unique structure, which is going to be specific to DNA, known as our double helix. And remember, they're going to run anti-parallel, meaning one side's going to go up, and then the other side is going to go down. They're being held together by hydrogen bonds, which are going to be very important because they're just strong enough to hold everything together, but just weak enough to be broken apart during DNA replication. So let's go ahead and test our knowledge and see exactly what we know. So just like previous lectures, I'm going to go ahead and give you guys a couple seconds to pause this, give this a try, and then we'll go ahead and check it out together. All right, and welcome back. So let's go ahead and check this out. So again, when it comes to nucleic acids, what we're going to have is going to be our RNA and our DNA. And number 2, circle the correct items in the sentence below. Chromosomes are primarily made of DNA. Remember, they're going to be found in the nucleus. And DNA is divided into regions known as genes. Again, there's going to be specific portions that are going to code for specific things. Adenine correctly base pairs with which other base? In this case, thymine. Remember, adenine A, apples, are found in tea trees, thymine. Cytosine correctly base pairs with which other one? Remember, cytosine is C, and it's going to bind together with our G. Remember, garage. Cars can be found in garages, or cars need gas. On one strand of DNA double helix are the bases right here. So we have T, G, C, G, A, T, T, A, C, T, T, C. So what is the sequence for the complementary strand? So one way you can think about it is this is one half or one side of it, and we're trying to find the complementary piece. You can see T right here is going to go with A. So remember, A always goes with T and vice versa. So looking at this first one, you can see that A is a T. So that means this is not going to be it. T, A, so that is going to bind together. T and T do not go together, so we can cross that one out. And then you can see T and A right here. So we move to the next one, G. We know C and G are going to go together, cars, garages. So we can see C and C here. Our next one is going to be C. C goes with G right here and right here. G goes with C. We have C here and C here. And then A goes with T, because remember, we're dealing with DNA. If this was asking for RNA, A would go with U. So A, again, goes with T. T, this one checks out. Right here, you can see that we have a U, meaning that this is not going to be correct. That one is meant for RNA. So this one right here would be the complementary set, or the one that matches up with this for DNA. And then we would do the exact same thing for number 6. So on one strand of DNA, double helix, are the bases T, G, C, G, A, T, T, A, C, T, T, C. And then which one of these are going to match up well? Again, same thing, where you have to identify what we're looking at first. In this case, it's going to be DNA. So we know DNA, again, is going to follow this base pairing rule, A going with T, C going with G. So we're just going to go ahead and find the complementary set, or the other side to this right here. So again, we would just do the exact same things. You can see we have T over here. T goes with A. So right off the bat, this can be crossed out along with this one over here. You can see we have A. Those all work. G goes with C. C over here, so on and so forth. And we'd go ahead and try to match these up with their complementary sets. In this case, the answer would be E. All right. And again, that is going to conclude our lecture for DNA. So I look forward to seeing everybody in the other lecture. Again, if anybody has any questions, comments, or concerns, please feel free to reach out and I'd be more than happy to help you out. All right. I'll see everybody in the next one.